Thanks, guys. My name is Dan, and I'm here today to talk to you about an idea that we borrowed from physics, stochastic resonance, and how it applies to bacteria that can resist antibiotics. So before I want to talk to you about stochastic resonance, I'm going to talk to you about the idea of noise, or stochasticity. So noise is ubiquitous in nature, and it's really important because it means that cells can experience variability both in their environment and inside their cell, inside a cell at the protein expression level. And cells can experience noise in space or in time. So these environmental conditions or protein levels can fluctuate. And what this means for biological systems, and when you're trying to manipulate biological systems, is that the more noise you have, the response of your biological system tends to go down. And natural systems where we see noise would be in biofilm-like environments or inside tumors, and this can make disease treatment really difficult under these conditions. And so we wanted to know how cells can exploit noise in their environment to try to address some of these problems. So there are two established ways that cells can take advantage of noise in their environment. The first is bed hedging, and this has been pretty well dealt with in the literature. The idea is that cells experience noise in the spatial domain, but not the temporal domain. So basically, they've got static variability, and if they can match their gene expression with the environmental conditions that they're facing, they can maximize the growth of the population as a whole. And this other idea, stochastic resonance, requires that the conditions inside the cell and the conditions outside the cell are fluctuating. So what is stochastic resonance? So stochastic resonance is the idea that as the noise in a system increases, the performance increases. And eventually, as you continue to increase the noise, you'll eventually see a decrease in performance, leading to this bell-shaped curve that we see right here. So stochastic resonance has been applied in prosthetics, where if you induce stochastic electrical pulses to the organism that's trying to use the prosthetic, you can improve the rate of learning and improve their ability to manipulate the prosthetic. And stochastic resonance has three main requirements. The first is a nonlinear system. So this is just a fancy term for a system that can affect itself through some sort of feedback. The second is a periodic signal. So this can be a signal that varies up and down over time. And the third is stochasticity, or specifically stochastic switching. So this is a kind of noise where a variable, in this case a protein level, fluctuates between high and low very quickly over time. So, given these three criteria, we wanted to build an experimental system where we could test whether bacteria that were antibiotic resistant could take advantage of stochastic resonance. And so to build a nonlinear system, we used bacterial cells that were expressing TEDx. Now, TEDx is a protein that degrades tetracycline, and when it degrades tetracycline is normally preventing the cell from being metabolically active and expressing TEDx. So in this way, production of TEDx affects its own expression, and you have a nonlinear system. Second, we grew these cells in a 96-well plate format, which can be periodically shaken, which can reflect periodic disturbances that cells might experience in, say, like a gastrointestinal tract or another environment like that. Finally, we need stochastic switching. In theory, cells in biofilm environments should stochastically switch back and forth with their gene expression. That concept can be represented by using viscous media to produce gradients inside that a cell might experience and then respond to by switching its gene expression. So because no one had actually proven that a viscous media would cause stochastic switching, we went ahead and did that for the first step of our validation of this system. And we took cells that were expressing or using the same promoter to express a marker gene GFP that they would normally be using to express TEDx, and we grew them in our viscous media. And when they grew, we quantified the GFP positive cells and the GFP negative cells that we saw in culture, and it was roughly 50-50. And then we sorted these cells out and regrew new populations from all low GFP cells and all high GFP cells. And we were trying to prove that cells that started low could end up high, and cells that were high could end up low. And we found that cells, no matter where they came from, were able to switch fates, suggesting that stochastic switching is going on inside this environment. So next, we took our cells that were resistant to tetracycline, and we grew them in a variety of media viscosities, so increasing the amount of noise that we see in this system. And as we increased the noise, we found that we were able to see a statistically significant increase and then a decrease in the level of the relative survival between the tetracycline-resistant bacteria and the non-resistant bacteria. And so this was exciting to us, but we wanted to have the correct controls to show that the bell-shaped curve that we see here wasn't a result of some other phenomenon that was going on in our experimental conditions. So we started removing the requirements for stochastic resonance and saw the subsequent effect on the system. So first, we got rid of the periodic shaking that we experienced in our culture, and we found that as we increased the noise under those conditions, we decreased the relative survival of cells. And then we eliminated the feedback by getting rid of tetracycline in our cell cultures, and we found that under all different media viscosities, cells grew exactly the same. And so this was very exciting to us, but then we wanted to test other regulatory strategies for antibiotic resistance, because constitutive regulation of antibiotic resistance isn't the only option. So we settled on two strategies, inducible and oscillatory control. And this is because inducible control is very well characterized for antibiotic resistance in the literature, and oscillatory control is representative of circadian rhythm-like behavior, which we're starting to observe more and more in microbes. So we tested a whole bunch of different conditions, but this is just the choices that we're showing you. And we did not see any statistically significant increase and decrease in the behavior of these systems. So it suggests that constitutive regulation is really important for seeing the stochastic resonance phenomenon that we observed earlier. So this is all very cool, but the core, the exciting part of stochastic resonance is that it can be applied to any system. And we wanted to think about how you could actually apply stochastic resonance to processes besides antibiotic resistance. And to really get a good idea of this, you need to know the fourth requirement for stochastic resonance, in which the periodic signal and the stochastic switching have to be synchronized. This is why they call it resonance in the first place. So knowing that there needs to be synchronization between these two processes is important, because if you're trying to maximize the effect of stochastic resonance, say you were trying to deliver a sugar to the gastrointestinal tract to try to improve the growth of a certain microorganism, you would want to modulate your delivery such that cells only get the sugar when they're able to accept it and able to process it. And likewise, if you're trying to do the opposite thing and deliver an antibiotic to a population of cells that's antibiotic resistant, you want to deliver it at the appropriate time where cells are susceptible to the antibiotic. And stochastic resonance provides a framework with which to do this. And so with that, I've shown that stochastic resonance can improve the growth of microorganisms under antibiotic stress, and we theorize that this can be used to improve the power, efficiency, and precision of drugs that we use to manipulate microbes and potentially other cells. And with that, I'd like to thank my lab, especially my advisor, Chimeng, and my lab mates. We had a student in the lab who goes by Bay, who helped characterize the oscillatory genetic circuit that we used in these results. I'd like to thank the funding sources for supporting our lab, and the DEB for supporting me during the Biotech Fellowship. And I'd like to thank you all for listening. I'll take any questions.